Well, in the first part of this lecture set, we talked about mitosis and we went through PMAT, prophase, metaphase, anaphase, and telophase. Now we get into the important part, for at least from a genetic standpoint. We're going to talk a little bit about meiosis. So here's where we've been. Okay, we've been talking about this fertilization event that goes on, producing zygotes, making them diploids, and that's where mitosis takes over, right? This concept of epigenesis, where we get this piece of code. The code is then used as the basis to form all of these new tissues, these organs and structures that we have. And it occurs through by cell division, by mitosis. We have these blastulas and all sorts of different forms that come along, 2-cell, 4-cell stages up to blastula and others. And then we get embryogenesis, the formation of buds and things, eventually leading us to a final organism, as we see. And that final organism, the coloration and the pattern and all the other things are determined by these basic genetic rules, right? What did you inherit from dad and what did you get from mom, and how are those expressed by the cell? Is it dominant or recessive? What is it? Okay. Now for this part, we want to concentrate on the top part here. Okay. I'm going to talk about the part that involves meiosis and leading up to, of course, fertilization, the random combining of these gametes, as they're called, to form the secondary product. Now, why? This is I think that's a valid first question. Why in the world do we have two different mechanisms here? Okay, so clearly reproduction can occur by mitosis. We can make new things, right? We can make things. But why do we do that? Why, in many species, of course our own, do we not do like the little hydra and just form a bud at some point and that bud break off, right? And why don't we clone ourselves? Maybe when you're 32 years old, all of a sudden a little growth comes out of the side and there's a little you walking along. Why do we go through all this other process? I mean, it's really dangerous if you think about it. Uh, if you're a prairie chicken, or in this case a grouse, in the spring you're out there and you're sort of cryptic and hiding most of the time, but when it becomes reproductive season, you feather up and you start flying around and flirting and making yourself obvious to predators. If you've been fortunate enough to be in the West in the springtime and late and early summer when the elk and things are fighting, they just really don't pay any attention to you. They just fight and often injure themselves. Or if in Africa, if you get there in maybe southern Africa in December or so, our winter, but their spring, you'll see the animals just on the side of the road, just males, fighting and fighting, just paying no attention to you, and often hurting themselves, sometimes locking horns and ending up not able to get loose, doing all sorts. Why do we do that? And then the act of reproduction itself. Transmitting sperm is difficult. If for fish and other things, you may just throw it into the water column and hope. Other things where you actually have an intermittent organ where you transfer it. And if you do have that, you can get all sorts of things happening with mammals and things. Humans, we have diseases, all this stuff going on, and the process of all that. So it's dangerous, right? And why in the world then would we invest so much energy and time into this type of reproduction when there's other methods that we could use? Well, it's a pretty simple answer. Pretty simple answer is that there's variation. Nothing creates as much variation as the process of segregation, independent assortment. We'll talk about recombination later on, but just those two processes. Don't have to worry about recombination at this point. Just meiosis, just going through the process of meiosis, producing segregation, independent assortment, makes more variation than anything. We talked about early on that variation is critical. All biological systems need variation, right? Systems change. If you clone, you're always basically the same. Unless there's mutation, everything that comes from this one thing is all identical. And so whatever can happen to one can happen to all of them, right? If you have variation, yeah, you don't get to give everything to your progeny that you have, but you can mix and match and you get this multiple form of variation. This is where meiosis comes in, right? We will go through the process of talking about meiosis and form, but we're talking about 2N, okay, being through replication, making 4C, going through G2, and then starting the process of meiosis, which divides us. Instead of 2 daughter cells that are both diploid, we're going to end up with 4 daughter cells that are in fact each haploid. Okay, so just kind of think about it. First, if you're going to have sexual reproduction and you're going to combine DNA, first thing you got to do is make a way to cut the DNA in half from each individual, right? I mean, if we were to reproduce by rubbing hands together, the somatic cells, of any type, we'd have a 2N cell and a 2N cell, we'd have 4N. And eventually we'd have 8N and 16N, and we just fill the cell up. So somehow we have to get this side to 1/2 to N and this side to 1/2, so that when they fuse, we end up back at 2N. So meiosis is a reduction. Okay, it's also the way we generate variation. I'll keep coming back to this. It makes sexual reproduction feasible. It makes it where you can overcome the cost and expenses of that, at least in a Darwinian sense. And it's, for us in genetics, it is the mechanism that gives us rise to both the principle of segregation and the principle of independent assortment. Okay? So just to remind you, replication and condensation, basically the same whether you're going through meiosis or mitosis. Okay, for our purposes, replication, we're going to make sister chromatids, we're going to make dyads, we're going to compress them and all that, just like you would in mitosis. Okay, now let's start. Meiosis is PMAT twice. That's about as simple as I can make it. Prophase, metaphase, anaphase, telophase, prophase, metaphase, anaphase, telophase. Although that second prophase usually doesn't exist or is very short. Okay, prophase, final shortening, thickening of those chromosomes and formation. Now, something very important happens during prophase 1 and metaphase 1, as we call it, the first time we go through the process, and that is that these homologs, which have replicated long ago, come along and they move around through what was the nuclear envelope area, and they eventually find each other and connect. And they connect at the centromeres like this. So a dyad, this is the original homolog from mom, maternal, original homolog from dad, paternal, replicated, and then by prophase, they start to move around and they start to connect. We call this a synaptonemal complex. They're said as synapse. Synapse means to connect. Okay. At this point, you have two dyads. This is a dyad. That's a dyad. These are sister chromatids of each other. So a lot of terms here, but dyad, di for two. When they fuse together, you got 1, 2, 3, 4. So you've got a tetrad formed. Okay, and they fuse up. They do it with what's known as the synaptonemal complex. Really cool structure. We're not going to spend a lot of time talking about it, but there are a lot of proteins involved. You'll learn more about it in cell and other areas, but a really cool connecting area that connects between these two dyads to make this thing called a tetrad. We're going to call it synaptonemal complex. Okay. Now we've got them hooked together. Okay. So when we go to metaphase, they go together. They can't. They, being the two homologs, are stuck together. They can't get like this yet. Okay. So they're going to travel wherever they are to the metaphase plate looking like this. So at metaphase 1, they line up across the equatorial plane, the metaphase plate, and our spindle fibers come out as they would before, right? And connect to the centromeres. But this time they're connecting to a centromere on this dyad over here and a centromere to this dyad over here, right? Very important. Look at the next slide. Just look at it for a second. In mitosis, these two dyads could care less about each other. They're off in whatever orientation. As long as they're on a central plane, they're fine. Okay. In meiosis, they're fused together. That's the big difference, right? Okay. That's what happens. All right. So we've got them done that way in metaphase. Anaphase, those little bungee cords pull apart. And when you do that, you separate the homolog. So whatever was maternally derived goes over here. Whatever paternally derived goes over here. That's the process of separation. Okay. Continues through anaphase. We keep moving forward. We get to tearing phase. We telophase up. At the end of meiosis 1, the first process through, prophase, metaphase, anaphase, telophase, okay, we end up with two daughter cells. And these daughter cells, we're going to say, are 2C. They're twice as much DNA. What they're twice as much DNA is that they're just duplicates of each one. So now you have this homolog that was replicated off over here. And this homolog replicated is off over here somewhere, okay? All right. Well, now what you're going to do is it goes back through the whole process, but this time you're going to separate these dyads independently. They're just off separate cells now, not in any real relationship, okay? Usually there is some short prophase too, but it's very quick and it depends on the cells and the organisms. But for us, metaphase 2 lines up something like this. Anaphase 2, we pull apart the dyads, right? And so you can always do this with your fingers, right? Even kind of like I'm doing right here in front of you. And then we get to telophase 2. By that point, we've broken apart the full cells. And each of these cells are called haploid daughter cells now because they're carrying in, right? They're no longer 2N, so we've got 4 cells. Each one has one of these chromatids, another one has this chromatid, one has this chromatid, one has this, but they don't have this, okay? They have something, they're separate, okay? Then, very cool, you can go through the spermatogenesis or oogenesis, production of eggs. Spermatogenesis or pollen production as well generally produces 4. So each of these cells that are haploid ends up being a sperm. Oogenesis, for the most part, produces just one large egg, with these other parts of it being sloughed off as things called polar bodies. You end up with one haploid egg here. That's why there's usually more sperm or pollen than there are eggs or ova. There's usually messages put in here and all kinds of cool things that are happening in oogenesis and spermatogenesis. And we're actually conditioning our DNA to send signals to our kids while we do it. But these are cool processes that we'll talk about at some other course, but they occur. And we now have, but it's the same process, right? For us, it's meiosis. Okay, so we've gone through meiosis. Now we can have fertilization. We can go back and we can do the whole thing over again, right? Meiosis has allowed us to take what was a diploid organism and produce these haploid gametes that can now fuse back together to make a diploid, right? So 2 of these gametes, one from each parent, fusing together will give us back, and we start the process then of epigenesis. All right. Now, what's that have to do with Mendel, right? What was Mendel's ideas? Segregation, Mendel's first principle. During the formation of gametes, these alleles separate so that each one, talking about the separation of homologs, talking about meiosis. When did we see that? We saw it in anaphase 1, right? Anaphase 1, the paternal DNA and the maternal DNA pulled apart. Big A, if it's over here, big A, because it's replicated, and little a, if it's over here, separated. Separation of homologs accounts for Mendel's first principle, right? That's what meiosis is all about, getting that DNA that you got from your mom and dad pulled apart. Now, what about independent assortment, the other one? Well, that gets slightly more complicated, okay? It occurs as well in meiosis. Everything that we're interested in as far as Mendel occurs in meiosis 1, right? And we generally say it's at metaphase 1, okay? Why? Because even though the physical action occurs at anaphase 1, it's already set up by metaphase 1, right? So let's look at it. Okay, let's take a individual with a genotype, big A, big B, little b. So dihybrid het, right? All right, let's take them and let's take an equatorial plane. Let's take a maternally derived dyad and make it big A, big A. Okay, the paternal we'll say has little a, okay, the black ones, right? Okay, when the next chromosome carrying the B locus comes in, right, we'll put them on separate chromosomes. When it comes in, it could line up with the maternal on this side and the paternal on that side, okay? If they do, right, then you're going to end up with gametes because you're going to separate right here, right, through the red line. You're going to pull big A dyad off and big B dyad off at anaphase 1. You're going to pull off little a, and little b at anaphase 1. And so big A, eventually, when you go through the second phase, you're going to split these dyads. Well, split these, you're going to end up with either big A, big B, or little a, little b. That's what you're going to get, right? That's through one trip. Now, we're not doing any crossing over recombination. We're simply talking about going through the process of meiosis. That's what you would get, okay? But we know that when you take an individual that's big A, big B, little b, there are 4 possible gamete types, right? There's big A, big B, little b, but there's also big A, little b, and little a, big B, right? Where do they come from? Well, you can't produce them in one trip through meiosis, okay? It won't work. You have to go through at least twice. That's one of the reasons we use fruit flies, or you may use corn or some other time. You have to see a lot of progeny because one trip through meiosis gives you one outcome, all right? You have to have another trip through meiosis. The reason is that the same genotype, the same dyads could have lined up like this, okay? Maybe this one lined up the same way. The maternals on this side, paternals on that side. But what about the other ones? Well, it's literally like taking those other chromosomes and just throwing them at the wall or throwing them at your screen. They land wherever they're going to land. So they could just as easily land like this. Okay. If that happens and you go through the process, there's only two types of gametes you can get. You're going to get big A eventually with little b and your final gamete product, or you're going to get little a with big B. Okay. So one trip through gives you one side of this or the other. One trip through meiosis. You have to go through at least twice and probably multiple times in order for it to really become 1/4 of the gametes look like big A, 1/4 look like little a, little b, excuse me, big A, big B, little b, or big A, little b, or little a, big B, 1/4, 1/4, 1/4, 1/4. Okay, it takes multiple trips. That's why you look at multiple progeny to see it. Okay, we don't see it in one trip through, right? This is the principle of independent assortment. Big A's are separating from little a's, that's segregation. Big B's are separating from little b's, that's segregation. Assortment is two or more segregation events thrown together, right? Well, here's one and there's the other. So we throw those together to get independent assortment. That's how we could get this outcome, all right? Now think about humans. Fruit flies are simple. They have 4 pairs of homologs, so there's a few combinations that you can get. Think about humans, right? Humans have 23 pairs of chromosomes, right? Think about all the possible arrangements, 2 to the 23rd power, essentially, somewhere up in the number of particles in the Einsteinian universe or something. So it is possible that you could produce an egg or sperm, whatever you're going to produce, that exactly had the DNA that your dad gave you, right? All the black things, all 23 could line up on one side, all 23 of your moms could line up on the other. That's possible, okay? Astronomically small probability that you could produce a sperm or an egg that's exactly what you got from either parent. Why? Because you got 23 chances and they're random as to whether they line up like this or they line up like this. Somewhere in that 23, you're going to get a flip. So they're not exactly what you got. And that's the variation part, right? We've now taken DNA from dad, DNA from mom, you got it, and then you mixed and matched it all up to produce your gametes. And each gamete type that you produce each time you go through meiosis will be different. So it's an incredibly important system for generation of variation. Okay? This is what drives independent assortment. All right, I want to show you the biggest errors that I see with students. I'll ask them to develop this idea of show me the relationship between meiosis and independent assortment, and somehow I'll get this written down. Okay, magically I'll get big A, little a. I mean, that's not possible, right? I mean, that would be like you taking a piece of paper and you put it in a copy machine and the English come out as Spanish or German or something. I mean, it's not going to happen, right? Copy machines and molecular copying, which is, replication is just a dumb system. It reads exactly what's over here and does its best to make a perfect copy. Now, there might be a mutation or something, but we're not talking about that. Okay, you're not going to convert English to German just by feeding it through a plain old copy machine. Nor are you going to produce big A into little a by going through the process of molecular copying, which is replication. So if you do that, you're going to get a big bleh, and people are going to be really unhappy. Okay, whoever's grading it, me or the computer, is not going to be happy with it. We want to see it you starting like this and like this. We're not doing any crossing over any of that at this point. Okay, we don't need that to have independent assortment. Now, it turns out that independent assortment can really help it sometimes, but we're not going to worry about that. We're not going to include that for the first part. Okay. All right. Well, that concludes basically everything that you're going to need for the discussion of Mendel and how Mendel relates to meiosis. May want to go back and review this one a couple of times. Make sure you look at the notes and play with this last part here. Play with trying to set up with different genotypic combinations. Try to set up a case where you do show that you get independent assortment.